Hello, my name is Phillip Kogan. I'm a high school student in Walnut Hills High School, Cincinnati. I would like to start by acknowledging my advisor, Dr. Meller, and my co-authors, Dr. Shamsi and Mr. Regal. So there are multiple problems with generalized medicine, which is the structure that we have used for hundreds of years for treating people. When a physician makes a decision about diagnosis or treatment based on the population average, he or she doesn't always take into account the patient's other characteristics that might rule out options available to the average person but shouldn't be available to that person. As a result, we prescribe non-optimal drugs and drug dosages that lead to internal problems which we could have avoided if we had selected an alternative drug. Additionally, it can be crucial to quickly select the best treatment options because there may not be time to try multiple options. So a solution to these problems would be precision medicine, which is an emerging healthcare model that relies on big data. We now have the ability to measure large amounts of data and store them for each patient, and we could tailor diagnosis and treatment to each patient's genome, habits, and other characteristics if we were to implement this technology. This would mean that a physician could select a viable drug and dosage for a patient, since he could predict how the patient would respond to different drugs and dosages based on his genome before testing different types of drugs. If these data were to be collected early on, we could also test for diseases that a patient is likely to contract and prevent illness as soon as birth. Certain types of cancer, for example, are almost curable if diagnosed early. So we want to characterize each individual patient, and for this we would want to know his data, for example, his habits, the environment he's exposed to, his genotypes, and how responsive he would be to different treatments. The first two we already know about because the patient knows what he does and where he lives. But the last two require obtaining his genetic information and then comparing it to patterns we learned from analyzing population data. Today we have the ability to obtain the individual's genetic information, but we still don't know how to use all of it because we are still studying the genome. So let's say we have a patient's genome, or at least part of it, and we want to predict the patient's type of a certain characteristic. For our study, we chose to predict smoking as an example characteristic because the patient knows for sure whether he is a smoker, so the data is reliable, and because there are available data with many smokers and non-smokers in the sample. So how would we create a predictor for smoking from this genetic information? First, we would select a specific group of people for our patient, for example, Caucasian male group, and download data about that group from a database. In our case, the database was CBioPortal. Next, we would modify that data as necessary for art to interpret it, and finally, to produce a decision tree which we can use to predict our patient's smoking status. So we've downloaded the data about the people in the same group as our patient, but this data contains thousands of gene variables, and many of them likely have little to do with smoking and would only confuse our predictor in a final model. So how do we select the interesting genes? A very simple way to do this is to look at how frequently the genes are mutated in smokers versus non-smokers, and then sort the genes by the greatest difference. This way we look for different behavior of the variables in smokers and non-smokers and select the most different ones. In our case, we selected the top 50 genes because the other genes would be filtered out by the decision tree algorithm later on. So now that we know which genes we are interested in, we can query data from the study with only those genes and download the response. Now this response data may contain more information than we need. For example, in our case, it contains the type of mutation. So we can map these values to just a normal or a mutated value for our basic tree. That way, our decision tree will split branches based on a mutation or a normal status. In the future, we could add multiple levels to our variables and expand our tree with this additional information if necessary. So then the branches would be split into amplifications, deep deletions, and other types of mutations. But for now, we're just going to go with a simple tree in which every gene is split two ways, either a mutation or normal. And now we're ready to make our model. So when we first create the model based on this data, we get a very large and unreadable tree that tries to use every gene in our list to fit our data. So the problem with this model is overfitting, so it probably would not be a good predictor for other data. And some of the leaf nodes contain very few samples, which is also not good for reliability. So if we tune the tree and rerun the algorithm with pruning, which is removing less significant nodes, and indicating a minimum leaf node size, we get something much more reasonable. And you can see that from all of those genes, actually only a few seem to have a connection with smoking. And now this tree can actually be used to predict something for our patient. So how can we improve the model in the future? Well, one way would be to consider that. So additionally, this process could be packaged into a single application for general use. The application would allow users to query their data, select important variables, and tune their predictor all from a single interface. Such an application would allow professionals who might not be familiar with R to use this tool in their own field. And for now, that's it. Thank you so much.